and this is a video on imaging anatomy of the basal ganglia. It's going to be a very short video on a very specific topic, and it's mainly aimed at medical students or radiology residents at the start of their neuroradiological training. But if you're an advanced neuroradiologist, well, you're welcome as well. Maybe there's something you might pick up or learn here, or maybe don't. Don't shoot me if you don't. So let's get started, and let's make it easy to get started. Yeah, when discussing the anatomy of the brain, I think there are three main components of the brain. And I'm hoping I'll be able to discuss each and every one of them with you in various videos. So what are those? Well, first of all, we have the gray matter. The gray matter, which contains numerous cell bodies, mainly of neurons, but also astrocytes, other cell types, and relatively few and mainly unmyelinated axons. And in contrast with that, we have the white matter, which contains relatively few cell bodies, but a tremendous lot of myelinated axons. And myelin is a fatty substance, as you know, and it is myelin, or this fatty substance, that is responsible for the white color of white matter on anatomical specimens, and that has given it its name. And lastly, we have some sort of cavities centrally within the brain called the ventricles, which are filled with cerebrospinal fluid and also contain the choroid plexus, the structure responsible for producing cerebrospinal fluid. So in today's topic, I'm going to talk about gray matter. And gray matter can be divided into two types of gray matter based on location. We have superficial gray matter. In most of the gray matter is superficial gray matter located along the outermost part of the brain and also referred to or called the cerebral cortex. And the anatomy of the cerebral cortex is something I have already discussed in another video, so look it up if you're interested. And then we have gray matter structures located centrally or deep within the brain. We get neurons that form clumps or nuclei, and these are called the deep nuclei. And the basal ganglia belong to the deep nuclei. So this is an example of an axial T2 weighted image, and if we zoom in on the frontal lobes, we can clearly discern the superficially located cerebral cortex, which has a higher signal intensity on T2 weighted images than the white matter located underneath it, centrally within the brain. And if we then zoom in on these structures located along the lateral ventricles and the third ventricle, we can discern here the superficially located cortex of the insula, but then centrally along the ventricles, we have all these structures that have a signal intensity on T2 that is very similar to that of the insular cortex and is not signal intensity of white matter. These are all deep gray matter structures. And there are several we can discern, and I'm sure you know most of them or have heard of them. We have the caudate nucleus located along the wall of the lateral ventricle, and at the level of the frontal horn, we have the head of the caudate nucleus, which is basically the biggest part of the caudate. Then we have here a lens-shaped structure called the lentiform nucleus, lentiform meaning lens-shaped. And then lastly, we have the thalamus located along the wall of the third ventricle. So let's continue. So let's continue once again with an axial T2 weighted image. Let's zoom in. So these are all gray matter structures located centrally within the supratentorial brain. And if we zoom in on these structures, we can discern the thalamus, once again, the lentiform nucleus, which consists of the putamen. So it has two components, the putamen, the most lateral part and the biggest part. And then centrally, we have the globus pallidus. And lastly, we have the head of the caudate nucleus against the frontal horn of the lateral ventricle. Then we also have a very small rim of gray matter tissue located between the lentiform nucleus and the cortex of the insula, which is called the claustrum, and the function of which is still a matter of a lot of debate and speculation. Nobody really knows what the claustrum does exactly, but it's there and you can see it on your images. So what are the basal ganglia? Well, first of all, the term basal ganglia is from an anatomical point of view not correct. A ganglion is a collection of neurons located outside the central nervous system. Well, clearly we are still within the central nervous system, so the correct term should be basal nuclei instead of basal ganglia. But hey, the name is there, everyone is using it, so we're not going to try and change that now. So it's just, one of those little things. The basal ganglia, strictly speaking, consists of a group of 3 pair nuclei, which together are sometimes called the corpus striatum. And what are these nuclei? Well, we've already seen them. We have the caudate nucleus, which is a C-shaped structure running along the lateral ventricle, and the most anteroinferior part is the head, also the biggest part, and then we have this very small tail, which extends all the way to the temporal horn of the lateral ventricle, and then we have the lentiform nucleus consisting of the globus pallidus and the putamen. I'm going to discuss these structures in a little bit more detail, but first a short word on the function of the basal ganglia. Basically, the basal ganglia have some kind of modulatory function. There is a circuit between the cortex and the thalamus, and the basal ganglia are located between cortex and thalamus. They receive the striatum, and here meaning the caudate nucleus and the putamen, receive a lot of input from the cortex, and then this input gets forwarded to the substantia nigra and also to the globus pallidus, which consists of an external part and an internal part, and also to the subthalamic nucleus. And all these connections modulate the activity the striatum has received from the cortex, gets forwarded to the thalamus, which forms a loop to the cortex, and we get some kind of continuous loop or feedback loop, whatever you want to call it. This loop is called the cortico-basal ganglia thalamocortical loop, and one of its main functions is that it regulates voluntary movements. So voluntary movements are generated by the so-called pyramidal system, and this system, the cortico-basal gangliothalamocortical loop, basically modulates pyramidal activity and is therefore called the extrapyramidal system. So when you have a disorder of the extrapyramidal system, you won't get a hemiparesis, but something goes wrong with the modulation or the regulation of your movements, and you get disorders associated with either too few or too slow movements, hypokinesia, or too much movement, too big movements, too speedy movements, whatever, hyperkinesia. I'm sorry if I pronounced that wrong. I'm not sure how to pronounce it. So hypokinesia and hyperkinesia. But that's not the only function the basal ganglia have. They are also involved in our reward system and play a role in the development of addictive behavior, for instance, and also play a role in the regulation of emotions, cognition, behavior, and so on. Basically, they can receive input from anywhere from the cortex, so they are involved in a lot of functions. This is a very strongly magnified T1 rated image of the basal ganglia. And what do we see? We see, once again, the caudate nucleus. And why is it called the caudate nucleus? Well, the Latin word cauda means tail. And it's a C-shaped structure with a big head and a very small tail, which extends all the way to the temporal horn. It has a role in regulation of movements, but when you get isolated lesions of the caudate nucleus, patients will mainly present with behavioral disturbances like psychosis, aphasia, abulia, and so on. Then we have the putamen. Putamen is the Latin word for shell, and maybe you see a resemblance with a shell if you use your imagination, or you can try to imagine that this is basically the shell of the globus pallidus, however you want to try and memorize it. And lastly, we have the globus pallidus from the Latin word pallidus, meaning pale or white. And why is this nucleus white? for the pathologist, but also for us neuroradiologists on the T1 weighted image, because it is traversed by a lot of myelinated axons, and these are responsible for the white color of the globus pallidus. Let's focus a bit on the caudate nucleus. As said, the caudate is a C-shaped structure running along the lateral or along the wall of the lateral ventricle with a very big head, a somewhat smaller body, and a very small flimsy tail, which extends all the way to the amygdala and the temporal lobe. If we look at the caudate on sagittal images and we zoom in a little bit, we can really appreciate the morphology of the caudate. We see here in black the lateral ventricle, then the caudate bordering it here in red. So this structure here is the thalamus, and this here is part of the putamen. But we are going to talk about the caudate. So this is the caudate head, the somewhat smaller body, and then here the tail would probably start, but we can't see it because I was not able to generate an image which visualizes the curved caudate nucleus in its entirety on just one image. This is what a caudate looks like on axial T1 weighted images. So if we slide somewhat lower, then we get here and here and here at the uppermost level. This is the body of the caudate running along the central part or the sella media of the lateral ventricle. And then lower, we have the caudate head, which borders the frontal horn of the lateral ventricles, also seen here on this even lower slice. Can you see the tail? Well, yeah, sometimes you can if you really look carefully and you try to find it. So let's see, this is the head of the caudate. I encircled it, and we're not able to see the course of the lateral ventricle on these coronal slices, but the tail would run along the lateral wall and then make a turn. So it would first turn to the back, then turn forwards again and end up above the temporal horn of the lateral ventricle. So if we zoom in on the temporal horn, we can clearly recognize the hippocampus. Then here we have the temporal horn of the lateral ventricle and the choroid fissure. And this very, very, very small gray matter structure is the caudate tail. So if you look carefully for it, and if you really want to find it, well, sometimes you can. These are T1 and T2 weighted images. And this is the lentiform nucleus or the lens-shaped nucleus consisting of the putamen and the pallidum. And I'm now going to talk a little bit more about the pallidum. There isn't much I can say about the putamen, but I can about the pallidum. If we look carefully at the pallidum on T1 flare and T2 weighted images, and I have encircled the pallidum in red here, we see that the signal intensity is different from that of the putamen and the caudate nucleus. It is somewhat more hyperintense on T1 weighted images and darker on flare and T2 weighted images. And the reason for that, I've already told you. It's because the globus pallidus is traversed by a lot of myelinated axon fibers, and that is responsible for the fact that the signal intensity resembles more that of white matter than that of cerebral cortex on your MRI images. If we look carefully at the pallidum on T1 weighted images, we see that the signal intensity is still somewhat higher than that of white matter. So we can make a distinction between white matter and the pallidum, and we can even see two white matter bands in and along the globus pallidus. First, we have a white matter band forming some kind of border between the putamen and the globus pallidus called the lateral medullary vellum. And then inside the globus pallidus, we have another white matter band called the medial medullary vellum, which divides the globus pallidus into an external part located laterally and an internal part located medially. And these two parts are not only anatomically distinct, but also functionally. They play different roles in the cortico-basal ganglia thalamocortical loop. And if you want to know what these are, go a few slides back or read a good article or handbook on the subject. So the term striatum, which is often used as a term to refer to all the three nuclei, caudate, putamen, globus pallidus. Some authors use it only to refer to the caudate and the putamen, and I believe that's probably the most correct, but never mind that. Anyways, the striatum derives its name. It actually means striped. And why is that? Because on pathological specimens, but you can also see it on imaging, there are a lot of gray matter bridges between the caudate head, or the caudate, not just the head, and the putamen at the level of the anterior limb of the internal capsule. And in Latin, these bridges are called the ponticuli caudato-lenticularis. And if we zoom in on this coronal T2-weighted image, we can really appreciate them. Here we have the caudate, this is the putamen, and then we have all these very small, it's very beautiful image, these very small gray matter bridges traversing the anterior limb of the internal capsule and connecting the caudate and the putamen. And I already told you, or maybe I didn't, I can't remember, but functionally, the caudate and the putamen have more in common with one another than the putamen and the globus pallidus. So the term lentiform nucleus is basically just an anatomical descriptive term, because from a functional point of view, there's basically no sense in lumping the putamen and the globus pallidus together. Makes more sense to lump the putamen and the caudate together. Okay, this is a sagittal T1 weighted image, and if we zoom in, and I like zooming in, we see here the putamen, this is the caudate, and then we have these very small gray matter bridges between the caudate and the putamen. I've already told you that the basal ganglia play an important role in the regulation of voluntary movements, but they also play a role in the reward system of the brain, and thus in the development of addictive behavior. And one nucleus specifically plays a very important role in that, and that is the nucleus accumbens. The nucleus accumbens is part of the so-called ventral striatum, and the putamen and caudate, or then the dorsal striatum, but that's not that important for this topic on radiology, is involved in the reward pathway. I already told you that. And can you see it on imaging? Well, not really. You can't really delineate it, but you can have an idea about where it is located, and that is in the antero-inferior most part of the striatum, basically where you see a large gray matter connection between the caudate and the putamen. So this is the caudate, this is the putamen, and you see that at the antero-inferior most level, they were already connected by the gray matter bridges I talked about. But there's also an immediate, pretty big connection antero-inferiorly, and here is the nucleus accumbens approximately located. So you can't really say, well, these are the borders of the nucleus accumbens, but you have an idea about where you could find it. Now, what structures are also part of the basal ganglia, not from an anatomical point of view, but from a functional point of view, and I'm referring once again to the slide on the cortico-basal ganglia thalamocortical loop, that are the substantia nigra and the subthalamic nucleus. The subthalamic nucleus is very difficult to directly visualize on MRI images, so I'm not going to show it or discuss it. Basically, it's located slightly above the substantia nigra. Slightly, but it's very difficult to really show it or delineate it as said. The substantia nigra, on the other hand, is often very easy to detect. These are susceptibility-weighted images, and if we zoom in on the midbrain over here, we can see some kind of, it looks a bit like a face with the big eyes in the middle. And this is the substantia nigra located centrally within the cerebral peduncle. And this here, the eyes are the red nucleus, not part of the basal ganglia system, but we can always see them at the level of the substantia nigra. So the substantia nigra is also often clearly visible on T2-weighted images, as we can see here, and this is the red nucleus. And if you look carefully on susceptibility weighted images, you can often see, let's magnify them a bit, that the substantia nigra has a shape that resembles a swallowtail. If you use your imagination or use this figure to put it next to the substantia nigra, and this is basically the normal situation. There were theories that the more hyperintense part located here in the substantia nigra corresponded to the so-called nigrosome 1, but this has been debunked or is a bit debated at the moment. So just forget about it. Just remember that this is what the substantia nigra normally looks like. Of course, you need really thin slices with a good angulation through the substantia nigra and of high quality to really appreciate the swallowtail sign. But if you see it, that's the normal situation. Absence of the swallowtail sign is associated with or seen in Parkinson's disease. And already the final slide. Let's talk about the white matter structures we can see at the level of the basal ganglia. What do we have? Well, let's just go from medially to laterally. This is the caudate nucleus, and here we have the thalamus, and then we have a white matter structure located between the caudate and thalamus and the lentiform nucleus, and that is the internal capsule. And the internal capsule consists of an anterior limb located, of course, anteriorly, and a posterior limb, and where those meet, that is called the knee of the internal capsule. Then, if we move further laterally, we have here the globus pallidus, then we have the lateral medullary vellum dividing it from the putamen, and the white matter band located along the lateral surface of the putamen as the external capsule. Then we have the very thin linear gray matter structure of the claustrum. Then we have the extreme capsule, and we end up in the insular cortex finally. And that concludes this very short presentation on basal ganglia anatomy. I hope you liked it. Of course, when you have a short presentation like this one, there isn't much you can conclude. So I'm just showing you a slide showing you the main deep gray matter nuclei we discussed here. If you have any comments, feedback, or questions regarding this presentation, please let me know. You can leave a comment in the comments section, or you can send me an email to neuroradiology.online@gmail.com. I always reply. I get a lot of emails, so I always reply, but don't panic or don't think I'm being impolite if you don't receive a reply right away. It takes me some time to reply to everyone. So thank you very much for watching, and stay tuned for more videos on neuroradiology.